So in chapter 7, we looked at the cell membrane and how things are regulated in getting into and out of the cell. Chapter 8 is all about metabolism. Metabolism, let's write down that definition, is the totality or the sum of an organism's chemical reactions. So if you look at just one cell, it's all the chemical reactions that occur in a cell, and there are lots of them going on at any given second to keep your cells and therefore you alive. And so the first part of this chapter, we're going to talk about chemical reactions, types of chemical reactions, and energy, because some chemical reactions require energy to occur. Some chemical reactions release energy. So we're going to look at energy and chemical reactions. And then the second part of the chapter, we're going to look at the substances that speed up chemical reactions. What are those things? What speeds up chemical reactions in a cell? Enzymes, right? And so the last part of the chapter is about enzymes, and so that's kind of how it's broken up. So, totality of an organism's chemical reactions. It says it manages the materials and energy resources of a cell. Oops. And before I move on from this, let me explain what this picture is showing you. So oftentimes there's usually a pathway to lead from your starting molecule. Your starting molecule is often called your reactant, right? And then your ending molecule, which is your product. So in this class and in many biology classes, it'll be written from like this. Instead of A to B to C to D, sometimes chemical reactions are shorthanded, written like this, where they just give you the reactant and the product, where this arrow represents many smaller reactions that occur to get to there. And so that's what this is showing you here. So this is a pathway. So to convert A to D, there are many smaller reactions. In this case, there's 3. And so A needs to be converted to B, then converted to C, and eventually you get your end product. And each one of those individual reactions are catalyzed or sped up by a different enzyme. So therefore, to get from A to D, you need 3 different enzymes to do that, and oftentimes we just write it like this. Let me give you an example of that. An example of a reaction that we skip a bunch of steps in between is the reaction for photosynthesis. So photosynthesis, I know everybody has learned this at some point. Let's do this reaction. So here's my arrow. So we're going to have reactants and products. Anybody remember what goes in for plants for them to use in photosynthesis? Or what's the main purpose? Let's start with there. What's the main purpose of photosynthesis, Colby? That's cellular respiration. It's the opposite. Anybody remember what's the purpose of photosynthesis, Derek?
To create ATP?
No. No. It actually has nothing to do with ATP. Okay? To make sugar, and that sugar is usually glucose, right? So I'm going to write glucose, formula for glucose. C6H12O6. All right, so there's our formula for glucose. So that's the purpose of photosynthesis. This is the main end product that we want to get out of photosynthesis. To make glucose, what does the plant use?
Water and carbon dioxide.
Yeah, it uses water and carbon dioxide. So this, more specifically, 6 of each one of those. All right, so these are what we considered your reactants. This is your product. There's one other product that's released from plants that is a waste product for plants, because this is the main purpose for photosynthesis. The other, O2. There's oxygen. So oxygen is released from the plant. All right, so that is our that's our chemical reaction for photosynthesis. This is how we usually learn it. This arrow, though, there's hundreds of reactions that occur between this and this. There's lots of things going on there in terms of that, and so therefore this would be an example of, these are our original molecules, these are our ending molecules, but then a lot's going on in between there. So this is a pathway. And so oftentimes we shorthand that. And so really there's lots of enzymes that are involved to get to your glucose and your oxygen at the end. Okay, so that's an example. All right, so let's categorize pathways. So we just talked about a pathway, a lot of reactions are pathways. There's two different types that you need to be familiar with. The first one is called a catabolic pathway. So what is that true about that? It releases energy and you're breaking down complex molecules into simpler molecules. So you have a more complex, end up with simpler at the end, and energy is released in that process. Example, digestive enzymes break down food and energy is released. Opposite of catabolic pathways are anabolic pathways. These guys consume or use energy to build complex molecules from simpler molecules. So we take smaller molecules, put them together to build something. And so the example is using amino acids to build what? What would amino acids be used to build? A polypeptide. I put a protein, all right, but a polypeptide, absolutely. So like a protein in your muscles, let's say, it could be anything. Okay, so now I'd like you to look at that other sheet that you picked up. It's actually from a biology coloring book. Well, we're not going to color it, I'm just going to go through it with you. And we're going to look at it and apply catabolic and anabolic pathways. Before I do that, that photosynthesis equation on the board, would photosynthesis be catabolic or an anabolic pathway? Photosynthesis. Is energy released or absorbed? Are you building smaller, taking smaller molecules and building larger ones, or breaking down larger ones and releasing smaller ones? What do you think?
Would it be anabolic?
It is anabolic. Anabolic, we just wrote, it uses energy to put smaller molecules together to build larger molecules. Glucose is the building of the larger molecules. It uses energy. Photosynthesis, what's the energy it uses? It's light, sunlight, all right? So it absorbs sunlight from the environment, absolutely. And so let's go through an example of the relationship between anabolic and catabolic pathways in our body and how they actually work together in our cells. So here you have this picture here. And we're just going to focus on this part of the picture right now. And so this is the cell membrane right here. And this is obviously inside the cell, outside the cell. And this is small organic molecules that are going to go into the cell and be used in one of two different pathways. So let me give you an example and relate it to stuff we've already talked about. So let's say you eat starch. So you eat starch. So it goes through your digestive system. What is your digestive system going to do? It's going to digest it, right? Well, when we eat starch, can you right now off the top of your head tell me what starch is? So because you're still going to be, it's a polysaccharide. You still need to know, this is from a couple chapters ago, but we still need to know what starch is. It's a polysaccharide. It's a chain of sugars. What sugar specifically? Glucose. So it's a glucose hooked to a glucose, hooked to a glucose, etc., etc., etc. All right. Where do we find starch in nature? Plants, right? It's the plant's storage polysaccharide. It stores glucose for later use. So potato plant, the potato itself is a bunch of starch from that plant, right? And it's storing that. So when we eat starch, most of you can tell me that, we're going to digest it. We're going to digest the starch. Can you tell me what we're literally doing to the starch when we digest it? Jacob.
We're breaking it down.
You're breaking it down. Be more specific. What are we doing to break it down? What's happening to the starch?
We're using enzymes to break the polysaccharide chain.
Yeah. When you say break the polysaccharide chain, what's breaking? Like, look at my picture here, what's breaking?
Bond. Glucose.
Yes. So enzymes are used to speed up this reaction. The bonds between the glucose break. When enzymes do that, they're speeding up what process? By what process did we learn about that when we break down molecules? Opposite. Hydrolysis. Hydrolysis is adding water to split molecules. Hydro, water, lysis to split. So we would add water, so enzymes help speed that up. So we're adding water to split the molecule. Instead of one large polysaccharide, now we have many small glucose molecules. So we have all these different glucose molecules. And so that's what these little guys here, let's say, are. So these are your glucose molecules. All right, so then your glucose goes from your digestive system into your bloodstream and then goes to all the cells of your body. And so this is one of the cells of your body. So glucose now needs to enter, diffuse, high to low concentration. That's why it's going to go in. How does glucose get through the membrane? To go through the lipid bilayer or protein? Protein. So therefore, I'm going to put a little transport protein here, and glucose now enters the cell. And that glucose is going to be used in one of two different ways. We can either use the glucose right away for energy. Remember, energy, glucose is used for energy, or we can store the glucose. All right, so let's talk about storing the glucose first. All right, that's this part here. So notice here this arrow, and you have all these different larger molecules. You have protein, starch, fat, DNA, phospholipid. Which one of these would glucose be used to make? Starch, right? Starch is only though in plants, right? If this is an animal cell in us, remember we store glucose, excess glucose, in our liver and our muscle cells. Remember that? And so if this is a liver or muscle cell, what are we going to store our glucose as? We're not going to hook it together to store it as starch. It's going to be what, Megan?
Glycogen.
Yeah, it's going to be long chains of glucose that we call glycogen. So this is in animals. So we can take those small glucose molecules, hook them together, and store them. And so this would be what kind of pathway? Taking smaller glucose molecules and hooking them together to make a larger molecule. Would it be catabolic or anabolic? Anabolic, right? All right, so I'm going to write here anabolic. This is the anabolic pathway. Now, I used the specific example of starch and breaking it down into glucose, but notice here we can do the same thing where you eat protein. What protein is a chain of amino acids. When we digest protein, it would digest into amino acids. Amino acids would come in and we can build proteins, all right? So quite literally, when you eat a cow protein is a chain of amino acids. You break the bonds between the amino acids just like we did with the starch, and you have individual amino acids that then come into the cell, and we put them together, and we use the amino acids that was once part of a cow protein to build a human protein, all right? And so then we can make human proteins that way, and so we build fats the same way, and so on and so forth. So this would be an anabolic pathway. Anabolic pathways need what to occur? Energy. So where does that energy come from? This is the second use for glucose. Glucose can be used for energy, and how we get energy from glucose is we break it down. It's quite literally the opposite of photosynthesis. And so if we read photosynthesis from right to left, that would be cellular respiration, breaking down glucose. We use oxygen and glucose and break down that glucose into carbon dioxide and water. So what happens is you take this glucose, which is C6H12O6, made out of carbons, hydrogens, and oxygens, and we break that down into smaller molecules. The two smaller molecules we break it down into are water and carbon dioxide. So we're breaking it, larger molecule, breaking it down into smaller molecules. In that process, energy is released. So I'm going to write here. Some of that energy is released as, it says here, H. That's heat energy. And so energy is released as heat. This energy is unusable. We as a cell, this happens in our cells all the time, we can't harness heat energy and make it do work for our cells. So some of that energy is released as heat, some of that energy is able to do work for our cells. So this represents the energy to do work. So when we break down glucose to these smaller molecules, some of that energy released actually drives reactions that need energy, these anabolic pathway reactions, so building glycogen and so on. But a lot of that energy is released as heat and is unusable. This is why when we break down a lot of glucose to get energy, we heat up. So think about when you use a lot of energy, like if you go running or you're exercising, you get hot, right? And then the reason why you get hot is because the breaking down of these molecules into smaller molecules releases energy that's allowing you to work out or to run, but a lot of the energy is released as heat, and that's why you heat up, okay? And so, so the purpose of that, though, is to provide the energy to drive those anabolic reactions, all right? So that's the relationship between those. So this, on this arrow, we should write catabolic. When I first learned this, I'm going to tell you the crazy way that I always try to think of ways for vocabulary to remember it. So catabolic pathways is breaking down molecules, right, to smaller molecules, and the energy is released. So it has the word cat in it. So I had a cat. I used to always think of my cat catching mice and breaking down tearing apart the mouse or the bird that it caught. So it's breaking it down. So catabolic pathways break down. So that's how my high school mind worked. So that helps you. And if that confused you, just pretend like that didn't happen just now. All right. So that's that. So let's go back to the notes. Okay, so those are our two different pathways. So now we're going to relate this to energy, because we just said that some of those reactions needed energy, some of those reactions released energy. So let's talk about the forms of energy, what energy is. So this should be a review. Capacity to do work. So there's two main types of energy we're going to look at that are important for this class. Kinetic energy, we've talked about this a little bit already with diffusion, energy associated with motion. We talked about that's really the reason why molecules diffuse from high to low concentration, because they have kinetic energy, they're moving around and they bounce around and therefore spread out, and that's how they get to equilibrium. So a form of kinetic energy is thermal energy, so heat is really that random movement of atoms or molecules. So I used the example with, the catabolic reaction just a minute ago, releasing heat. And so that's unusable energy for a cell. So heat energy for us is unusable energy. It's the most random form of energy that living organisms do not harness to do work. So the heat is usually passed off from us to the atmosphere, all right? And so potential energy is stored energy as a result of position or structure. So can somebody think of an example of something that may have potential energy because of its position? Colby?
Talk about last chapter with the membrane and depending on the concentration gradient and create potential energy against the membrane.
Yeah, absolutely. So having a high concentration on one side and a low concentration on the other side. Absolutely. Anything else? Think of it. Megan? Yeah. So water behind a dam, all right? And so when that is potential energy, when it goes through the dam and flows down, all right, and flows through the dam, then that potential energy can be converted to other types of energy and energy available to do work. We can have it do work. So this potential energy stored behind the dam, it has energy that water has potential energy because of its location. All right, if it was sitting in a pool, just in a lake, it would not have as much energy as it does stored behind the dam. How about structure? You might think of something that has potential energy because of its structure.
Would ATP be an example? Because as soon as you break one of the bonds and get ADP, you're releasing energy in the breaking.
Yeah, absolutely, absolutely. So molecules can have potential energy because of its structure. So like glucose has more stored potential energy than either water or carbon dioxide. And so, so that would be an example of that. So that type of potential energy is chemical energy. So this is how much energy is in the molecule that is available to be released when that molecule reacts with something. Okay, so broken down or whatever. And energy can be converted or be transformed from one form to another. And so that's all called energy transformation. So all of this should be a review. So we can convert chemical energy to mechanical energy to electrical energy and so on. And so those are energy conversions. And so this shows you just a simplistic, everyday kind of scenario where we see this person having different types of energy based upon their location. And so the diver up here has more potential energy standing on the platform than that diver, once they're done diving, has in the water. So has a greater potential energy, and then that potential energy is converted to kinetic energy as he dives, and then has less potential energy than up here, and then the kinetic energy is being converted as it gets back up to the platform, back up to potential energy. All right, and so that is an example, a very simplistic example of that. Okay, so let's look at specifically this thing called thermodynamics, looking at energy transformations or transforming from one type to another. So thermodynamics, the study of energy transformations that occur in nature. So, first off, things can be either closed or open systems. So closed system, I give an everyday example, in a thermos, the liquid in the thermos is supposed to keep your liquid either hot or cold, all right? And how it does that is it isolates what's inside that thermos from the outside environment. So closed system, there would be no transfer either of energy, so no heat is escaping from your thermos, or no liquids, no matter is escaping from your furnace, and so there's no interchange between inside that thermos and the environment. That's a closed system. Open systems are exactly the opposite, where energy and matter can be exchanged between, let's say, so you have a really bad thermos if there's energy being exchanged. So if you put something hot in your thermos, it's supposed to be a closed system, and an hour later it's ice cold, you don't really have a closed system there, right? All right, and so that would actually be an open system. All right, so living organisms, do you think they're open or closed? Do we exchange energy and matter with our environment? Yes, we do. We would die, all right, very quickly if we did not. So organisms are open systems where we're consistently exchanging energy and matter. So we take in our food, that's matter, and potential energy in the food. So let's look, oh, question.
What about like a sponge? Would that be considered an open or closed?
Open, yeah, because there's nothing closing it off from it. Yeah. So, so there are two laws that we're going to look at with thermodynamics. If you've had physics before, you probably have talked about this. If you haven't had physics before, that's okay. All right. First law of thermodynamics. You guys have probably heard this before. The energy of the universe is constant. So the quantity of the energy is always the same. But energy can be transferred and transformed, so it can be transferred from one substance to another, transform types, potential kinetic or something like that. But, so the type of energy isn't constant, but the amount of energy is constant. And you have probably heard this, that energy cannot be created nor destroyed. You've heard that before, right? Yes. All right, and so that's what we're getting at. So you can't create it, you can't destroy it, so it's always constant, it just changes forms from one form to another. Another word for this is the conservation of energy. And so those are two interchangeable words for that same concept. All right, so second, I forgot the little picture of the bear. So this picture of the bear, so energy is going to be is constant. This bear is going to get energy from the stored energy, chemical energy in the molecules that are part of the fish. And so then that bear is going to, what is it going to do with that chemical energy, that stored energy? It's going to break those molecules down. It's going to release energy, and that bear will have energy to do what a bear does, whatever, he walks around and so on. All right, so the second law of thermodynamics, this one you may not have heard before. Every energy transfer or transformation increases what we call the entropy or disorder of the universe. During every energy transfer or transformation, some energy is unusable, often lost as heat. So we've already talked about that. So when we take glucose, when we eat something that has glucose and we break those bonds of the glucose molecules, some of that energy goes to do cellular work for us. A lot of that energy is released as heat. That heat goes up into the atmosphere and so on. And so we went from having energy that was very orderly stored in this molecule to that energy being converted to energy for cellular work, but also heat energy. So what we've done by breaking down glucose is we've increased the disorder of the universe, because what has happened is we took something very orderly with this stored energy. When we broke it down, some of that energy got released as heat. That's the most disorderly type of energy you can have. And so therefore, after we've broken that down, the universe was more disorderly. We've increased that. And so that's what this little guy is doing here. So this is the bear running. Remember, he just ate the fish. He broke down the molecules, the chemical energy. When you break down glucose, you get carbon dioxide and water. That's what this is doing here. Some of that energy is being used for the bear to run. A lot of the energy is being released as heat. So now we have an increased entropy, all right, in the world. So the part of the energy that we actually are able to use to do work, so it's really basically the energy minus the entropy, is what we call free energy. Free energy, part of the system's energy available to perform work. So I'm going to introduce you to a new mathematical equation here. So the free energy, change in free energy, delta means change, during a process. So the change is final minus initial. So what? So the change of what happens before to after a process. So related to the change in enthalpy, this is probably a new word for you. Enthalpy is the change in the total energy. So these guys go together here. That's the enthalpy, it's the total energy. And a change in entropy. Entropy is represented by the letter S here, all right? And so let's look at this, what this equation means. So you have this equation, delta G equals delta H minus delta T. And T is the temperature in Kelvin, which remember is your degrees Celsius plus what? 273. All right, so let's look at this, delta G. So what this is the energy available to do work. So what do we have to do? So what we do is we take the enthalpy, or the total energy. So this is the total energy in the system, total energy. And we subtract the unusable energy. So That's the entropy, the disorder. So that's, if we look here, delta S, this is the entropy. This is disorder. So random disorder we're going to take away because cells can't harness heat to do work. Notice here though that this is multiplied by T, and T is temperature. If you think about this, if you increase the temperature, what happens to the randomness of the movement of molecules? It increases, right? So do you see if this number goes up, if the temperature goes up, then your disorder goes up. If temperature goes down, disorder goes down. So temperature is directly related to entropy, and that's why it's in this equation, all right? So we take all the energy available minus all the randomness, and we get energy, that the leftover is energy available to do work, all right? And so, we're gonna come back to this in a little bit here. So if we have a generic equation, A plus B gets converted to C and D, so those are your products. So this right here, it says initial state to final state. So before the reaction, this is your initial state. This should be right underneath there. This is your initial, and after the reaction, this is your final state. So what we do when we say delta G, delta means the change, and so we're going from GI, or that's supposed to be a little sub I, and GF. And so what we actually get is, to get the change, is how much free energy or energy available to do work after minus what was originally there. Okay, so we're going to come back to this in just a second. We're going to build up here. So let's look at this free energy, and so the energy available to do work, but it's also a measure of a system's instability, its tendency to change to a more stable state. Most things are working towards being, chemical reactions occur because molecules will go to a more stable state. So during a spontaneous change, free energy decreases and the stability increases. And we're going to go through an example of this in just a second. And equilibrium is a state of maximum stability. So if you reach equilibrium, it's the maximum stability. The thing is though, if our cells and all of our reactions reached equilibrium, the thing is that no work can be done. And so actually for living organisms, being a state not in equilibrium is the best scenario. So a process is spontaneous and can perform work only when it's moving towards equilibrium. If it reaches equilibrium, no work can be done. So let me give you an example here. Did I go too fast? Yep, I did. Gotcha. All right, so there's three scenarios here. Gravitational motion, where it goes back to the diver on the top of the platform, versus diving and eventually on the ending in the water. So that would be the end. This is diffusion, pulling in from last chapter. So you have a high concentration of the molecules, and then here it's evenly spread out. And then we have an actual chemical reaction where we see here a larger molecule being broken down into smaller molecules. Looking at all three of those, what I'd like you to do is, if the top is the initial and the bottom is the final state for each of those three, which one of those, the ones at the top or the ones at the bottom, have more free energy? Take a minute and think about that. Which ones, the ones at the top or the ones at the bottom, have more free energy? Another way to say that is more energy available to do work. Think about that for a second. Actually, talk to the person you're sitting next to. Figure it out. See if you can figure it out. Which one has more free energy?
Not talk about that, but earlier this year, like last year,
All right, let's come back together. More free energy, top or bottom? Does anybody want to share what you came up with? Nobody wants to share? Melon?
We thought that it might be like the initial, because it has more potential.
Yeah, absolutely, that's right. All right, so these guys have, so the G initial is a greater amount of free energy. These would be G final. All right, that's at the end. This is how much free energy would be at the end, and that would be less. All right, yes, because they have more potential energy. That's right, stored energy. And then, and so, next question. Which ones are more stable, the top or the bottom? Which ones are more stable, the top or the bottom? Talk. All right, come back. Who wants to take that? What do you think, Megan?
This is the bottom one.
Is more stable?
Yeah.
Absolutely. So this is more stable, and this is less stable. And so this is what we wrote about, that things are working towards reaching equilibrium. So like in this example with the diffusion, where you have a higher concentration versus equal concentration, of working towards equilibrium. So living things rely on moving from a less stable to a more stable state. And so in this case here, if we look at this and look at the delta G, so we just talked about the change in free energy, energy available to do work. Delta G here is, we figured that out by final minus initial here. Which one's higher, final or initial? Which one's going to be a higher? If we actually had a number, which one would be a higher number? The G, GF, free energy, GF or GI? Which one's higher? For this one, which one's greater, the amount of free energy in the final state or the amount of free energy in the initial state? All right, initial, right? And so if this was a number, we'd actually have a higher number, and this would be a lower number. So if we took a lower minus a higher number, what kind of number do we get? This is negative, all right? And this is going to lead us into the next thing.
Will that always be? It'll always be negative.
For this, hold on, all right. Let me get to the next slide. So let's look at this next slide. So free energy, the system's energy available to do work, and the change in free energy is GF minus GI. There are two kinds of reactions. First one, exergonic. In chemistry, you remember, you heard of a reaction that sounds a lot like this. What was it? No? Exothermic?
Yeah.
Okay. Yeah, that means releasing heat. All right. This is releasing any type of energy. Okay. So that's what the exergonic reaction, generic term for a reaction in which energy is released. These reactions are called spontaneous reactions, where energy is released. And the delta G is less than 0, which means that it's a negative number, right? That's a negative number, and so that's what we just did on, so those examples we just looked at the previous slide. When we took the GF minus GI, we get a negative number. What does that mean? That the initial free energy is higher than the final, and you get a negative number. So if you want to put that in your notes, I don't know if you can see it here, but if you want to rewrite that just so you can have it here to look at again, I'll just do that real quick. So initial is higher number than final, and so you get a negative number. which means then it is less than 0. And this is always the case for an exergonic reaction. So to answer your question. Okay. So an exergonic reaction, this is always the case. So any reaction that releases energy will always have a negative delta G. Yes.
Do you have a reaction with 0 as a final solution?
No. Then it really wouldn't be a reaction. She wouldn't be changing. Yeah. Usually it either releases some or. For endergonic reactions, the opposite of this, energy is required. So you have to have an input of energy. So you're absorbing free energy from the environment. That's where the energy comes from. And in this case, delta G is greater than 0. So it's going to be a positive number. And so if we did this same thing, I don't have an example of this, but based upon what we just learned, which one would be higher? G, the free energy in the initial state or the free energy in the final state to give you a delta G that's greater than 0? Yeah, the final. So it's going to have more energy. So what you end up with, so you're at the beginning, you have less free energy. At the end, you have more free energy. But remember, energy can be neither created nor destroyed, so that energy has to come from somewhere and it's absorbed. So, and that's going to give you a positive number. Question?
Why would anything ever become less stable? Like, doesn't that mean that whatever it is becoming less stable because it's got like a positive number?
Not necessarily. So when we take glucose and hook them together to make glycogen, it necessarily. Well, I guess it would be. It depends upon how it's put together, the bond angles and things like that. So just because something is being built doesn't necessarily always mean that it's less. But yeah, you're right that because there's a benefit to the living organism to use those. So because we have to have that to break down to do other, you know what I mean? So that's part of where if we reach equilibrium, we would never be able to do anything. So therefore, we have to make less stable things so that we can in turn break them down. All right, so we will stop here.